Thank you, Philip. It's great to be here and to see so many people joining. My name is Emma, and I'm an application scientist at LUMICKS, and I'm happy to share today what I think is the beautiful thing that we can do with our system. So we're going to dive a little bit deeper into dynamic single molecules analysis and what it is, and then we will go into the tools that we have and how it works. And the majority of the time we will spend on the science that you can actually do with it, because I think as a scientist, that's the coolest thing. And finally, we'll wrap up a little bit on how LUMICKS as a company supports scientists in their journey to discover breakthroughs. So a really quick introduction. We are LUMICKS. We are a company that is founded in 2014. We are based in Amsterdam. We have a lot of young, excited former academics and other brilliant people working in our team. And we have developed a technology which we call C-Trap. And we have already more than 100 systems worldwide, of which one actually in Israel, in Tel Aviv. And these instruments have produced over 130 publications to date. Actually, I think we're already at 140. Their publications keep coming up. So it's really a big contributor to the science. So let's dive into what it is all about. So the title of the talk is Dynamic Single Molecule Analysis. Why do we care about dynamic single molecules? We strongly believe that anybody who wants to study diseases or understand or even cure diseases or prevent diseases needs to look at the molecular level. That's where something goes wrong that can then cascade into either a regional or even systemic phenotype. However, studying something at the molecular level is not very easy because, as you can see in this animation of one of the many biological processes that are happening in your body, many molecules work together, tightly regulated in time and space. So you have two molecules coming together, doing something, bending some DNA or doing something else, and then they drop off and then another one comes. So this is a very dynamic process. These molecules, they interact together and every molecule does something different, but it can also be doing something different a few seconds later. So it's a bit of a challenge to really look at the dynamic molecular level. There are beautiful techniques that help us study at the single molecule level. For instance, we have really amazing techniques such as cryo-EM, which can look at molecule structure and composition to the angstrom level, and that's absolutely brilliant way of learning how a molecule looks like and where the functional components are. However, the downside of these techniques is that even though you get really high resolution, really single molecule, even single atom resolution, it's always a static image. The sample preparation does not allow you to really look at the molecule in a dynamic way. At the other end of the spectrum, we have beautiful experimental assays that can determine the function of a protein. For instance, live cell imaging, you really see a protein locating from one part to the cell to the other part of the cell. But often in these either bulk assays like gel assays or imaging assays, we need to image or detect a lot of molecules at the same time to be able to surpass this detection threshold. And the downside of that is that maybe the majority of the molecules that you are labeling or that you're looking at are not doing a very interesting function and might obscure those that are. At the same time, you are in any case looking at an averaged value, which may or may not be representative for the actual function of the molecule. So our system and our technique actually tries to or successfully tries to bridge those two ways of looking at mechanisms by really looking at single molecule in a dynamic way. And I hope that maybe now it still looks a little bit abstract, but at the end of the presentation, you will understand, you will get a sense of what we mean with that. So what is this beautiful tool that allows us to have the best of both worlds? This is called the C-trap. And basically it is a combination of optical trapping with fluorescent imaging. And this really allows you to have two types of outputs that you can fully correlate. We have fully integrated microfluidic system to build up your assay and to make your life really easy and to really quickly change experimental conditions. And finally, we have our software called Blue Lake, which makes all of these components, controls all of the components. You can automate all the components, and this not only helps you really in your workflow, but you also be able to correlate all the data outputs that come from the system. So let's look a little bit into optical traps, because this is something that typically optical trapping and optical tweezer setups were reserved for people who had at least a really good background in physics, if not a PhD in biophysics, in order to build a system. And we want to make it accessible for everyone. So I'm going to introduce you a little bit how it works. I will not go too much in depth because it's not necessary. And if you want, we can definitely talk more about it, but I could talk for hours then. So Arthur Ashkin won a Nobel Prize in 2018 for his discovery that you can trap particles, physical particles, with light. It relies on the principle that if you have a particle, in this case, a polystyrene bead, that's what we work with. If you hit it with a light particle, a photon, the difference in refractive index from the particle versus the surrounding water, for instance, causes that light will be refracted. It will be bent off its course, and because this is a bend, it will actually create a momentum the other direction. So basically, because the light particle is being pushed away, it pushes the bead the other direction. Now, if we have a focused laser beam with lots and lots of photons coming out of it, and a lot more in the middle than on the edges of the beam, we will be able to trap a particle in X, Y, and Z in our microscope. So that's the basic principle behind optical trapping. You have a focused laser beam and a particle will be trapped in it. So that's the physics behind it. We have a microscope objective that will generate this focused laser beam, and of course a lot of optics in the system that we don't have to worry about. And this is basically how it looks. If we have a camera and we are looking into if we have our focused laser beam right where the cross is, then a free floating bead will automatically be sucked into this trap. So you can imagine if you have two of those, you can put stuff in between and start stretching whatever you have in between. And that's basically the principle of what we are doing with our system. So how do we trap two of these particles? This is where our microfluidics also comes in. We have our microfluidics flow where we have beads, and we turn on our lasers and the beads will just fall into the traps. We don't have to do anything for this. We just have to turn on the flow and this will automatically happen. Now, these traps, these lasers that have these beads in them, we can move them freely. So this allows us to actually move the beads and stretch whatever we have in between. And not only that, we can also sense this tension that is between the two beads. But if we have two beads, like here, we zoom in a little bit, and there's nothing between these two beads, then if we were to plot on this graph to the right, the distance between these beads versus the tension or the force sensed on these beads, then of course it would be a flat line, because no matter how much you move them apart, there is no tension, nothing is pulling on these beads. However, if there is a piece of DNA, for instance, in between, DNA is rather elastic. So when it's still very loose, we will not see anything, no tension. But as soon as we reach the length of the DNA, then, and we keep stretching, then we will sense this tension. And this is exactly what happens if we stretch DNA. So you will see that the curve will go up. And this is really the tension that is sensed on the beads. If you look at the left bead, you can actually see that it's being pulled while the right bead is moving away. And this displacement we can sense, and then we know exactly how much tension there is. DNA is a perfect substrate for this, because we know exactly how DNA is supposed to behave. But we don't only have these optical traps. In our diagram before, I showed you we combine this with imaging. Now let's say that we combine this with fluorescence imaging, and we now don't have our DNA labeled, but we have proteins that bind to the DNA labeled. So we have two optical beads. We have our double-stranded DNA unlabeled, so we don't see it when we make a fluorescence image like this. And we have our single molecules that are fluorescently labeled, and we can see them sitting on the bead. Now, this is of course already extremely cool, because seeing an individual molecule or maybe a dimer binding to a DNA strand, that's already very, very impressive. However, we like dynamics. So what we prefer is to actually make a movie of it. And now we can actually see the proteins scan along the DNA, move along the DNA, and really see what their behavior is, because maybe their diffusion is dependent on its surrounding. So this is where we combine basically our optical trapping with our imaging. We can make these beautiful images of our proteins that we are literally holding between our extended hands. Now, in order to get to this assay with the DNA between two beads, we are using our microfluidics. The microfluidics consist of a few syringes in which we put our content, and then we have automatic valves that we can program or open and close manually, and it all goes into a flow cell. The flow cell looks like this, although normally you don't have this nice colors because your buffers are probably just transparent. We put food dye in here to really illustrate the principle of laminar flow. Because we introduce a pressure on the syringes, the flow of the liquids will be constant. There will be a constant flow through the flow cell of these liquids with different contents. And because this is a constant flow, the contents of these channels, of these syringes, do not mix, even though the flow cell, the lumen, is one big open space. So you can see here that we have beautiful colors, and they do not mix into something, I guess, brownish, if you mix all the colors together. And we can play with this, because if I now start the movie, you will not see something happening immediately, but trust me that the flow is going. If we then start to close certain channels, you will see that the other liquids will take up that space. So I'm gonna start it now. Soon we will close the red channel. There we go. And then we close the yellow channel, the middle one. And you can see that the green and the purple really take up that space. If we open it again, it immediately takes up this space again. Now, this means that if we have our particles trapped, we can move freely between these channels and expose our assay to different surrounding. We can use these channels to build up our assay, but also to change experimental conditions. I hope that that is clear. If not, feel free to pop in a question. So that's basically what the system is made of. So we have our traps, we have our imaging, and we have our microfluidics, and we have our software that helps us control all of this. Now, what the cool thing comes now. What can we do with this system? You can imagine if you have the principle of having two tiny particles that you can move freely around, there's a lot you can do. We have classified about 5 segments. Of course, there's much, much more, and I will go through the majority of these. But as I said, the possibilities are basically endless. So the first one that we will dive into are DNA binding proteins. So we have already seen a little bit of that. Basically, this is the field of research where everything that is focusing on the processing of DNA is can be grouped under. So whatever is happening to the DNA, transcription, translation, repair, editing, everything, if there's a protein binding on your DNA and doing something, this is your segment. If it's not your segment, we will probably hit yours later. So DNA binding proteins. In order to build up the assay, we use a very simple connection of streptavidin and biotin. So our DNA is biotinylated, and then we have streptavidin coated beads. Now, we can start with just looking at the mechanics of the DNA itself. I mentioned before that DNA is very well characterized, this is therefore very nice to study to confirm your hypothesis as well. So because only one strand of the DNA is biotinylated and therefore attached to the beads, if we start to stretch the DNA, we will change the structure of the DNA. And at one point, if we stretch it enough, maybe the 2 strands will actually melt apart. If we have low tension, say 30 piconewton, and we use a DNA intercalator such as Cytox, we can coat the DNA strand that we have between our beads. So this is how it typically looks. I think it's already extremely cool to look at, because how often can you see an individual DNA strand with your own eyes? But of course, this is only the starting point. If we were now to stretch the DNA up to, let's say, 70 piconewton, which we know is called the overstretching plateau, so really the DNA is having trouble staying together, we can see that indeed the DNA started to come undone, to really separate the 2 strands by the blue part. So the blue, we are using a protein that binds specifically to single-stranded DNA, and as expected, the 2 parts that were attached to the beads are now single-stranded. So that fits with that only one of the strands was attached, so it's easy to detach. But we can also see a tiny nick in the middle. In the middle of the green part, there is a section of blue. So this indicates that there was probably a nick in the DNA there. So if we then start to stretch, this could also be an origin of the DNA peeling, basically. So basically what we can do by just stretching the DNA, we can change the structure. We can even generate single-stranded DNA. And of course, this is already quite cool, but we like to see how proteins interact with DNA. And therefore, the structure is, of course, also very important, because maybe the DNA structure dictates how the protein behaves. And one beautiful example is the experiment with Cas9. As you probably know, Cas9, let's see if it runs or not. Cas9 binds specifically to target sequences, and there cleaves the DNA so that recombination can happen and gene editing can start. So we definitely don't want Cas9 to bind on off-target sequences, because there it could have side effects and damage your DNA where you don't want it to be. So in order to validate whether the Cas9 principle is suitable for therapeutic purposes, we need to make sure that this off-target activity is at least well documented and preferably prevented. So this video, which I really like, we have two optically trapped beads and a DNA tether in between, and we have one single DNA Cas9 molecule at low force. So this is beautiful. We would say, yes, we like Cas9. However, if we stretch up to 50 piconewton, we see a lot more binding events that are clearly not on the target sequence. And this is very reversible. Now, what happens when we stretch the DNA strand? This is where we change the structure of the DNA. So apparently the structure of the DNA really dictates how the protein binding behavior is. And this changing of this DNA structure is happening in your cell as well. So this is very physiologically relevant because in your cell you have constant supercoiling, positive, negative, opening up the DNA. So to mimic that under control is extremely valuable, but we can do more than this. So now we know that there is a relationship between stretching and the amount of binding, but we can quantify this. And for this, we like to take what we call a chymograph. Basically, we take only a confocal line scan through the DNA because we know where it is. It's always in the middle. And then we plot this over time. So per time point, we have one line scan. And if we then assemble this into a whole array, we basically get something like this. And now you can see all these green lines. And each green line is a protein binding to the DNA, staying there for a while because time goes on, and then detaching again. So we can calculate a lot of things from here. We can get a lot of binding kinetics, like the K on, K off, the duration of binding, diffusion. Cas9 is a pretty static binding protein, so it doesn't really travel along the DNA a lot, but if it were, you could see it by a diagonal trace, and then you can get diffusion coefficients and things like that. But moreover, because we have two of these red labels on this DNA that we introduce, and we know exactly on which base pair they are located, we can even determine the binding localization. So we can calculate, okay, at this spot, this base pairs, we see a lot of binding. Is there maybe a sequence there that could justify why there is a lot of off-target binding here? Is there maybe a very high matching sequence right there? And what if we would change this sequence? So you can get a lot of quantitative data from these images. Moving on to another segment, which is slightly different, which is protein folding. Of course, proteins can bind to DNA, and that is extremely interesting, but sometimes we are also just interested in the protein itself. How proteins fold is quite an important topic because many diseases stem from proteins that are misfolded. There's whole protein, whole classes of chaperones that resolve wrongly folded or aggregated proteins. So how proteins fold is essential to their function. And when that goes wrong, you might end up with disease. So studying protein conformation is very interesting. We can do this really nicely as well. How do we go about that? We can put a DNA strand in between, but we can basically put anything in between. So we can also put a protein attached to DNA handles in between. Now, this drawing is absolutely not to scale. Of course, your protein is a lot smaller, but just envision that you have basically two anchor points on your protein, and there you have a DNA handle, and these DNA handles, they are attached to your beads. So it's nicely suspended between those. Now, if we were to stretch this construct, we might trigger an unfolding event. Let's say we have the N and the C terminus linked, then we could stretch it to a linear chain of amino acids, right? But also you can design it differently. But in any case, you can really trigger this unfolding. You don't have to wait for it to maybe change conformation by itself. We can just rip it open. So how would we analyze this then? Here we are not necessarily looking at fluorescence in first instance, but we are really plotting the tension, so the force sensed on this construct. So if we look at this graph, we can see is what we call a typical force distance graph. So the x-axis is the distance. So the distance between the two beads, we can see that by our cameras. And then the y-axis is the force. So the force that is sensed on the beads. So basically the tension on the construct. Now, if we were to start pulling, the right bead is moving away, we would see this stretching that is typical for the DNA handles. They stretch in a certain way, but then we would see a sudden drop in force and an increase in distance. And this is literally that we have a protein that unfolds, and therefore our whole construct becomes a little bit longer. So an increase in distance, and because we suddenly have a little bit of extra length, we also have a little less tension. So the force goes down a little bit. This is a typical protein unfolding curve. Of course, if you were to go backwards, you would see it jump back again when the protein refolds. So you can do lots of these FD curves, force distance curves, to characterize one individual molecule. And you can, from these kind of graphs, you can derive several things. For instance, you can get the mechanical stability of a protein. Which force is required to unfold your protein is very protein dependent. Actually, it's even dependent on its surrounding. It could be that you, when your pH of your buffer is different, it's really harder to unfold your protein. And thanks to the microfluidics, remember the different channels, we can bring one protein from one channel to another channel and expose it to different surrounding. So the mechanical stability under specific circumstances can really be quantified. At the same time, if you have one drop, like in these two graphs, you know you have one unfolding region. But you can imagine that if you have a more complex protein that has lots of subregions that follow, that unfold sequentially, your graph would look a lot different. So you would see multiple steps with maybe different sizes reflecting the different regions. So the amount of intermediate unfolding states, as well as the length of the domain, you can determine from this graph. But because we can actively hold these beads and therefore the tension on the construct, whichever we want, we can also look at the kinetics. So basically what we are doing now is we're holding them at a pre-tension. And this is, in our case, if you look at the top graph, exactly at that point where the protein unfolds and refolds. And basically now the protein can be in either the folded form or the unfolded form. Both conformations it can be in. And if we just keep it there, and our system is extremely stable, so we can keep it there for minutes and minutes and minutes and like up to hours without any drift, we can see how quickly it shifts. Is it really a dynamic that it needs a very low energy barrier to keep shifting back and forth, or will it stay in one conformation for a very long time and then it unfolds and then it refolds again? And how is this affected by its surrounding? Because we can again bring exactly the same protein into a different channel where maybe an inhibitor or another small molecule is present. So we can, by just using the force sensing capabilities of the system, determine already extremely detailed characteristics of your protein in different circumstances. But it wouldn't be us if we don't try to also include fluorescence, because that's extremely useful as well. You can, of course, also introduce an intramolecular Fred pair. A Fred pair basically emits light, the acceptor light, if the two fluorophores are very close together. So if you put them on the domains that are supposed to be close together when it's folded, but will be put apart, if you're unfolding it, then you would be able to say with the fluorescence level, not just something about, yes, this protein is unfolding, but you are actually saying, yes, it's unfolding. And when refolding, it brings back these two components. These two parts of the protein are being put in close vicinity again. And if we do these experiments, then we indeed see in the bottom, you see this dynamics in the two states, the folded and the unfolded state of the protein based on the force readout, so the tension readout, and the top graph, which is looking very, very similar, is actually the fluorescence readout of your protein. So there you can really see, and not just in a one-dimensional direction, yes, there is changes in tension, but also in a potentially third tertiary dimension, that it's refolding, bringing two domains together. So there, for this section of the protein folding, I think this is an extremely added value to existing techniques as well, to really have something on the tertiary structure. Moving on to phase separation, because lots of proteins don't necessarily act on their own. We already saw that they act on DNA and maybe have cofactors, but also in the last years, we have realized that proteins like to aggregate together. And one example is, for instance, in the case of where on DNA, you have local condensates of really high concentration of proteins to do a specific function on the DNA. So for instance, you have a condensate of nucleic acid and of transcription factors and other components that are really required in a high concentration to do a specific function at a specific site, thereby preventing that the whole nucleus has to have this high concentration of factors. And in this experiment, they actually coated the DNA with a protein and then allowed the DNA to relax and see if it would just, start to become floppy, or whether it would really, the protein would trigger the DNA to really condense into a small, like a small blob. And if we look at this movie, you can see that the beads are being brought close together, thereby relaxing the DNA, but that the DNA will still stay in the middle. It will not start to become very loose and go out of the field of view. And there is a clear white dot in the middle that is really this accumulation of DNA that is really coiling up on itself with the protein there. In another paper, we have also studied the sequence specificity of these condensates on the DNA, as I mentioned before, where we have maybe a transcription factor at a specific promoter region where it condenses or recruits all kinds of cofactors it's needed. Here you really can see that, so this is basically a chymograph from top to bottom, that when we progress in time, moving down in this graph, that the foci or the condensation, the condensates on the DNA, the horizontal DNA, are becoming a lot more pronounced, really showing that they're accumulating proteins and also that they're very fixed in their location. And because we know the DNA that we have put in between, we all can also verify which genomic position these condensates like to get stuck on. And finally, because we know that, well, the principle behind optical trapping is that you have a different refractive index between your particle and its surrounding. If you have phase-separated condensates, they also have a different refractive index because they're phase-separated. So we could also trap them directly. So a lot of people who are studying phase separation and liquid-liquid phase separation, and they want to know how liquid-like are the droplets that they're seeing. And one of the characteristics that you can measure then in our system is the ability to fuse with other droplets. Now, knowing that we can trap them directly, we can also bring two of these condensates together and basically make them fuse. And the rate with which this happens says something about the liquid-like properties of your droplets. And this is a comparison between three conditions in which the concentration of pegs are changed as basically as a way to show the aging of the droplets. And if I play them now all together, you will see that the top one fuses pretty quickly. Plop, there it goes. The bottom one, the second one goes as well, but the bottom one, it really does not want to fuse. So how their fusing capabilities are, you can really see by eye. And of course, there's a lot of forces that happen here as well. So you're constantly measuring those as well. Moving on to the last segment of or last section of our talk, I'm combining basically two segments because I think there is some overlap as well. Basically, we are now moving a little bit away from individual molecules and more looking at a little bit of a bigger picture. Because, for instance, in the cell, there are more than just DNA and proteins, right? We have a cytoskeleton and we have motors running over it. And if you have two particles and you can stretch anything in between, why not something of the cytoskeleton, right? So we can also do that. As long as you can stick it to the bead, you can put it in between and start stretching it. In this example, they have put an intermediate filament, vimentin, between the two beads. It's a very small filament, only 8 to 12 nanometers in diameter. We can absolutely see this between our beads. And if we then start to stretch it, so here, this is a force strain curve. So it basically means that the different colors reflect the different speeds with which we are stretching. And you can see that there's a big difference in if you stretch a filament quickly or whether you stretch it slowly. And this says something about how complex is this structure and how much rearrangement is required to accommodate for this length increase that you are triggering. And of course, we relax all of them constantly, again, to be able to have multiple repeats. So you can basically put all kinds of structures in between your beads. And if you want to go even more complex, you can also study how motor proteins are walking on, for instance, microtubules. But there's more to the cellular structure. There's also the membrane, and the membrane is very dynamic and is very important for cells, but are sometimes very hard to study. So having a model system for membrane research that you can really easily manipulate is something that is being very valued by researchers. And one of these researchers that is extremely important in this field is Raya Sorkin from Tel Aviv University. She has a CTRAP as well. And she's doing these kind of experiments. These are actually her and her drawings. And what she is studying is, what she does is basically she coats 2 beads with a membrane that has membrane proteins, and then she brings them together, and the membranes will touch and they will interact, and then she pulls them apart. So you get curves like this, that basically you have a force, and it's 0 because nothing is really happening, and then you start pulling, and then suddenly you have a rupture. The protein lets go of the other bead, and then you get this spike. And then she does it again. So here you can see there is like 6 repeats, and you can measure the interaction force of this protein. But of course, you can do a lot more. Membranes are typically bilayers, and they fuse. But they can also do hemifusion, in which only one of the layers is actually fusing. So if you have a constellation in which you have 2 beads with 2 membranes, one actually has fluorescence label and the other does not, you can do an experiment like this, where you bring them together, and then you make these membranes, or at least the outer ones, fuse. And then you can see the fluorescence molecules disperse over the 2 beads. So basically the dark bead, which did not have any fluorescence, can only have gotten its fluorescence from the membrane fusion of the other bead. And this diffusion coefficient says something about the mobility of the molecules in your membrane. How diffusive are they? And finally, one very interesting paper that Raya has recently published. She actually reconstituted membranes from cells that she induced to make vesicles. These vesicles, which are membrane, of course, mainly membranes, she then used to coat complete beads. And in her case, she had one that really was showing the ACE receptor. So this is a COVID paper, very relevant, of course. The ACE receptor, and then the other vesicles were the ones who had the spike protein on their membrane. And then she brought them together to see the interaction between these 2 components, so between the ACE receptor and the spike protein. Now, of course, in this drawing, you see one interaction. You optimize your concentration so that you either have only one, or that you at least have a sense of how many interactions you have. But then you can do a lot of cool statistics. You can, for instance, determine the probability of interactions. So how many times do we have to approach in order to get an interaction? And you can do all kinds of combinations. You can have different beads with different membranes and see how that differs, but also the rupture strength. So if they interact, how much force do we need to put? How hard do we have to pull to rip them apart again? And in her case, she really sees that there is a huge affinity match between the ACE receptor and spike protein, much more than with any other combination or with an antibody that she uses. So this becomes immediately really relevant for fields like immunology, and also the more cellular oriented people. So with that, I would like to emphasize once again that this was just a really quick overview of a few of the examples that we have. We have a lot more, but I don't think we want to sit through a 6-hour webinar. I want to emphasize that there's much more that we can do. So in your field of research, probably if it hasn't passed by now, there's still a lot that we can talk about and we can consider. And we are happy to do so. We will also be visiting Israel in July. So if you want to know more about what you could do yourself, pop in a question or drop us a message and we can meet one-on-one. To wrap up, we of course realized that it's very nice to talk about experiments because they're nice and published and exciting, but there is a lot more to it than doing experiments. And know that at LUMICKS we are very aware of that, and we have basically support along the entire way. So yes, we have a team of expert scientists like me and my colleagues that can help you with trainings and with designing your experiments so that you have the best data that you can get. But at the same time, we also have support in sample design and preparation. We have actually a store where we have all kinds of constructs, and they keep coming. Our team is constantly developing new, also on request, new constructs to help you do your science. So we have DNA constructs with labels or with sections of single-stranded. We have kits where you can put any sequence you want in between. And we also have, of course, the tools to put your protein between your beads. We have an entire series of kits for that as well. Of course, there's also data analysis involved. For that, we have analysis software. We also have a community platform where a lot of scientists share their experiences and their knowledge and their scripts, even their samples. And finally, of course, the system will not break down, but if anything would happen, we have a very well-staffed service and tech support team that their main focus is to make sure that you can do your science. So we cover the entire range of things that you could encounter with having such a beautiful system. The adoption worldwide is very high. We have over 100 systems in the world now since 2016, when the first system got installed. And the papers that come out are, first of all, very many, and they come quite fast, and many from the systems as well. Like you have about a paper a year. And the most interesting thing is that the average impact factor of a paper with C-trap data is actually around 15. And that's a lot higher than in general, the average impact factor, which is, I think, around 3. So this really signifies that C-trap data adds something to your story that is being extremely valued by journals and by other researchers. And finally, yeah, we feel that bringing this technique to all the researchers, not just the physicist or the biophysicist who can build a system themselves, opens up so much more great science. So the user-friendliness of the system is for us a key component. And this is reflected in the fact that we have the all-in-one software, analysis software, and also all the biochemistry kits that are constantly being expanded so that you can really focus on experimenting and not necessarily on sample preparation. Because we really feel that your success is our success. We are a young company. The technique is extremely established, but researchers are brilliant and are constantly pushing the boundaries of what we can do with the system, which is definitely something that we celebrate. And helping you push those boundaries with our tools is also extremely advantageous for us, but also we enjoy it a lot. That's what we do it for. So really, your success is our success, and we hope that we can bring a lot of success in Israel with this technique. I'm happy to answer any questions now. I'm thanking you for your attention. See, I'm making a big, almost full hour. Feel free to also take a print screen of these contact details. As mentioned, Yair is the first contact person in Israel, but we are always happy to, if you're around Amsterdam, to show you around. The door is always open. As I mentioned, we are also planning to travel to Israel in July. So if you would like to talk to us in person, then definitely drop us an email as well.